Okay, today we will talk about recombinant DNA. What is recombinant DNA? Recombinant DNA is an artificially made DNA strand that is formed by the combination of two or more gene sequences. Steps of making recombinant DNA. The first step, isolated gene of interest. Second step, insert gene of interest into plasmid DNA. The third step, put recombinant plasmid into coli by transformation. The fourth step, plate cells into agarose gills containing apelin. The fifth step, expands the positive cones to exponentially replicate copies of desired DNA. The sixth step, isolated DNA. The seventh step, and this last step, test biological properties of your gene of interest. And this is how it works. The uses of recombinant DNA. Knowledge of the sequence of DNA segment has many uses, and some examples follow. First, it can be used to find genes, segments of DNA that code for a specific protein or phenotype. Second, homologous DNA sequences of different organisms can be compared in order to pilot evolutionary relationship both within and between species. Third, a gene sequence can be uh, secreted for uh, fun- uh, functional regions. In order to determine the function of a gene, various domains can be identified that are common to proteins of similar functions. Aims of recombinant DNA: to isolate and characterize a gene, to make desired alteration in one or more isolated genes, to return altered genes to living cells, artificially synthesize new gene, altering the genome of an organism, understanding the hereditary disease and their cure, impro- uh, improving human genome. The end. Thanks for listening.